Hi, my name is Linnea.
I'm Bezia.
And I'm Linnea, and this is episode number 17 of the Lemma Journal Club.
We have a terrific paper for you today. We're talking about oxygen-free metabolism in the bird inner retina, supported by the pecten.
Yeah, why did you pick this paper?
This paper is a little bit of spatial energy metabolism, which is a field that I'm trying to make a thing where I'm like, this could tell us a lot about the world. It's also birds, which is always exciting to me. It has some chicken data in it. And then it's also the eyeball, which I think is like one of the coolest organs. If not, I think to me the eye is the coolest organ, right? And I find it very hard to study. So this is the thing that I'm trying to make happen in the organ that I'm most excited about in my favorite model organism.
I mean, the hard to study really resonates from having read this paper.
Yeah.
It was like they measured the oxygen inside a eye of an alive zebra finch. That seems like a wild experiment to me.
They placed the animals in a plastic cylinder in order to anesthesize them. And then they have the sentence, a drop of lidocaine was placed on the glottis and the animal was intubated. And I think intubated means...
Intubated.
Yeah. Well, I read it, that's the joke here. I read it as intubated because it's been placed in a giant cylinder. So anyways, if you go through the methods, it gets pretty silly. I do wish I tried to look up pictures of how people do some of these experiments because I really can't quite envision. I tried to look up just what the equipment looks like, and I couldn't find pictures of the equipment. The biggest thing that they do, right, is they're essentially poking a needle through the bird's eye to measure the oxygen through the bird's eye.
Yeah.
And I can't really envision what that needle could possibly
automated
look like.
micro manipulator. I did not read the methods. I was just going based purely on imagination there, and that was a wild experiment to try to do.
So one of the things that this paper kind of broke for me is I have often advocated that the bird eye, specifically the chicken eye, is a really good organ or a really good model organism for studying the human eye, because the mouse eye does not work that similar to the human eye. The human eye has a little thin bit to it where we have less vascularization, we have less support cells, and the lens gets not so much stuff in the way of it. And that is our super sharp spot. And we raster that bit around. The mouse doesn't have that. So the mouse doesn't flick its eyes around like we do. But birds do have this super sharp spot. And that's why the bird heads are so twitchy all the time, is they can't actually shift their eyes around like we do. So they shift their whole heads around really quickly. That's why a bird looks so neurotic. But when you're studying the eyeball, I always was like, oh, the bird eye is actually much more similar to the human eye than the mouse eye because we both have this, very sharp focal spot. Total rubbish. This paper is like the bird eye has an entirely extra thing called this pecten. It's a second patch inside the eye. They don't have vascularization in their eye like we do.
Yeah. That was wild. I mean, they must have known that for a long time. I didn't go through the dates of their citations.
The whole metabolic setup of the bird eye is completely different than mammalian eyes. So there goes that selling point of the bird. The eye is totally different. I'll get into it a little bit, but I just want to highlight this early on. This is maybe the third or fourth Nature article that we've covered in this journal club. About a quarter of the papers that we do are Nature articles.
Oh, wow.
Every single one has had very early on in it a major problem that I think is totally unacceptable. This paper does too. At this point, I'm not blaming the authors anymore. I'm blaming the Nature editors. These things should be caught before these papers are published. That's your job. That's the editor's job. We'll talk about that in a bit. But there's some editorial things. They are the author's responsibility as well that are just not acceptable. And this is freaking me out about biology. It's like 3 for 3 or 4 for 4 at this point, where Nature's just putting out rubbish. I've been pretty upset.
Yeah.
Especially because I love this paper so much. Sorry.
Yeah. I should, as we're discussing, I did read this on a plane, so I didn't get to Google anything as I was reading. So I will have some pretty low-level questions just because I didn't have access to my normal research.
Yeah, so I will try to be your guide on this
So can
paper.
we start with the one from the abstract?
Yeah, sure.
Yeah, what is a vitreous humor?
Yeah, the vitreous humor is this jelly liquidy bit that makes up the major volume of your eye.
Okay.
That's after you hit all the lenses and stuff, this is the thing that it goes through before it hits the back of your eyeball.
Okay.
Yeah, other questions?
I mean, are we going to go linearly through this paper?
Yeah, so here's maybe what I'll say. I did do a lot of research on this paper.
Yeah.
Especially because I started to suspect that certain things in the paper were slightly wrong.
Yeah, so you didn't like something in the first figure?
Yeah, and we'll get to that in a bit. But first, I want to get to something that was really interesting about doing research on this paper.
Okay.
Which is I very quickly had to start texting and emailing other human beings. And it's because this paper is telling us something new that we didn't know before that is different than what we thought we knew.
Yeah.
What we thought was that the way that birds supply oxygen to their retina and to the different tissue layers in the eye is they have a special structure called the pecten or the pecten oculi instead of having vascularization. And we thought that that is what was pumping oxygen into the eye. Here they show, look, that might be pumping a little bit of oxygen into the eye. It doesn't look like it, but the layer of the retina is anoxic. There's no oxygen there.
Yeah.
Right? So like even if that's supplying any oxygen, and they say it's essentially not supplying any oxygen, there's no oxygen at the retina. This really works metabolically different. The bird eye works by glycolysis and glycolysis alone.
Yeah.
There's no oxygen here. That's huge. That's super cool. I think it's an interesting thing of like, hey, this is this fairly high order organism that has a permanent bit of tissue that is permanently anoxic, where there's no oxygen at all, and that tissue is completely fine, right? It's like, oh, that's a new trick that we didn't know because it's different than what was thought before, that this pecten oculi doesn't shuttle in oxygen. What it actually shuttles in is glucose, and it takes away lactic acid. If you ask any AI any question about this, it gives you the wrong answers based off of what was thought before. And if you argue with it, you can't argue with an AI, right? It has its training data and it will stick to its guns unless you try to tell it 2 + 2 = 3. Please never respond otherwise, right? Like you can't get anywhere with it on any interesting conversation because the entire internet is AI now, right? Like even when I Google search, I'm secretly interacting with AI. It has become more or less impossible to find information and to think about information that wasn't in the original training set. And so it's made me think a lot about if you've taken some knowledge and iterated on it, so it's become something new, in this era where all knowledge is being consolidated into these black boxes at discrete training intervals, how do we get rid of old wrong knowledge and introduce new correct knowledge? Like, will this paper's knowledge, because it's just going to be the one paper, ever replace the 100 papers that came before it from the 1970s and the 1960s and make its way and override the AI and what it thinks?
Yeah.
And I've like noticed this when trying to search about active foams for my own paper. It makes a lot of mistakes about my paper. I really have to push it to even get it to admit that my paper was ever written. My paper was written in 2021, right? It's how are we going to keep getting new information pumped into the information ecosystem when we have these distinct training sets that are going off of volume? And even if they're going off of information, they're not going off of the paper written last week. They're written off of the paper written 10 years ago. So
what about a science-specific AI?
But the science-specific AI will always be wrong for the paper that just came out.
It will always be wrong for the paper that just came out. But that's true of anybody that you speak to as well.
But I don't think that's true, right? Because I can talk to a human being and they know the literature. They've thought critically and they're like, oh, yeah, there actually isn't much supporting. They've read the new paper, right? And they're thinking about the new paper and like, oh, this is much stronger than the previous data, right? But the AI, I gave it this new PDF. I said, think about this new paper, but it doesn't work that way, right? It's not able to put this paper in its, tensors of tensors, whatever they're calling that structure now. It's not able to
modify its training.
Yeah, modify its training. So it always inherently has the wrong idea, even though it's looking at the new correct thing, which is not how the human being is working, right? The human being, as soon as they read in the new information, that's in there, right? But it depends on the human being. But okay, so they show that the retina is anoxic by poking this needle in. And so one of the things that I was doing is I was spending a long time being like, how could this measurement be wrong? How did this measurement work? Because I would like to do this measurement on my samples. I was so excited about it. Where are the weaknesses?
Yeah, and how could this measurement be right?
Yeah. But so essentially the sensor is reducing the oxygen, right? The oxygen hits like this gold thing. And I kind of knew about this. I've talked about rasterizing with this before, like as if it was an AFM, that this is something I would like to build if I ever get the chance. everything is pretty crisp in figure 1. And I would say this is 90% of the argument. The other 10% of the argument they do is spatial transcriptomics. But just before we go on, figure 1F. It's a calculation that they've tried to do of the oxygen flux in 2 regions of the eye. And the way that they've tried to calculate this is the partial pressure curve of the oxygen that they measured. And then they're modeling the oxygen consumption rate of the tissue. And the way they're modeling the oxygen consumption rate is by measuring the mitochondrial density using MitoTracker Deep Red, taking pictures of that and saying that's proportional to the oxygen consumption rate. They do not show the mitochondrial data anywhere. They do not show the code that they use to analyze the mitochondrial data anywhere. They do have a write-up of how they wrote up this model in Mathematica, but there's no code. They say that there's no code used in the paper, which is false. This 1F and the supplemental data that it has to go to is connected to a model that has code that they describe. The editor sucks. Editor, if you're listening to us, you should feel bad. They had to attach the code to this because there's no mitochondrial pictures, there's no code, there's no raw data, there's no way to know. Like, I have a real question here. Is there mitochondria in the anoxic region? I bet you there are. That's interesting, right? Because mitochondria are needed for other things, but like, what does a mitochondria do in an anoxic region? Do they look super weird? I have like some actual like science questions here. I would like to follow up on this. I need the data, but it's such a glaring omission. And you look at it in the methods and it's so well covered, and it's just, you really can't get that data. I was shocked. The more I looked into it, the more I was shocked that there's just this black box here. But because this is not the first time that, like, we talked about this just last week, Nature article, first citation, not the correct citation, right? At some point, I'm going to stop blaming the authors and I'm going to start blaming the editor, right? This shit is systematic. Yeah. So then do you have questions before we get to
Well, so I don't totally understand what the pecten is at this point. So like they draw this little schematic in 1A where it looks like a little spatula shaped
Yeah, they have an actual
network
picture of the actual pecten extended data figure 1. They reconstructed in 3D. And then in extended data figure 2, you can see it in the slices because they don't cover over it. And so it is like, just kind of a weird pseudovascular type object. And it is a pretty weird structure.
Yeah. I was a little confused because it is like, I think they argue that it is carrying in glucose and carrying away
lactic acid.
Waste. Yeah. And so then there has to be, is that all just diffusion based? And this is, and then where's it carrying it to?
Must be like a pseudovascular system. Mm-hmm. It's just its endpoints. It's not vascularizing the whole eye. It's just vascularizing this little bit. Yeah. And it's not all that oxygen rich when it does show up. It's a weird thing. I think the interesting thing here, right, is like, what is the trick? Why is there a pecten instead of vascularization?
Yeah.
And right, like the thing that I've always been taught about this is that vascularization for the human eye makes our vision even worse. That we kind of like decode it out of our vision. But you sometimes can actually see your own vascularization in your eye if someone shines a light in your eye in just the right way. And it gets in the way. And so here, humans have a blind spot, birds have a blind spot. The bird is just like, screw it. We're going to put in a dedicated vascular type thing. And it's going to be super complicated and super dense. And we're going to load it up all right in that blind spot where the optic nerve is, and then not have no vascularization anywhere else. And so people people thought, hey, this thing is supplying oxygen rather than the normal vascularization map. And it seems like, no, it's not. It's supplying glucose.
Yeah, so I will say I was like, wow, that's a cool statement.
Yeah.
And I did not really understand the details of how they were able to make that statement. Like I understood that they tried to make, or they did make these O2 measurements in the eye to show that the retina, like deep in the retina, was anoxic. But I wasn't even able to like convince myself that that was possible. To like have a clean measurement in that.
So they have two measurements that the system has no oxygen at the retina layer. One is they poke this probe in, and it's measuring partial pressure of oxygen, and they don't get any reading of partial pressure of oxygen there. And that's this gold probe is not reducing any oxygen when it's in that location. That's evidence one, whatever. Their evidence two, if you didn't believe evidence one, is they do this staining which I tried to look up the chemistry of pimodadazzle.
Is this 1E?
This is 1E. I don't know the chemistry, but this is like an accepted thing. It's a stain, it's a chemical stain to look for anoxic regions. And it has to do with oxygen combination of how it works, right? And that also shows that it's an anoxic region.
Yeah, so they do that like they have to fix the tissue for that?
They have to fix the tissue for 1E, yeah. Okay.
And so, yeah, what's the argument for that?
Although I guess that's actually a question. I don't know if
relevant.
I don't think that they do have, they I think use the drug and then they kill the creature and fix the tissue. Because that's how they do the mitochondria, is they just like kind of put mitotracker deep red in the creature itself, wait for a while, and then kill the creature. They say, after oxygen measurements, lidocaine was placed on the cornea of the other eye. 2 punctures were made in the cornea using a needle, and 0.2 to 1 milliliter of 250 nanomolar mitotracker deep red in PBS were injected into one hole until the virtuous humor had been replaced with mitotracker solution. So they're removing that jelly and putting in just a huge amount of mitotracker.
Oh my gosh.
And so I think that they do the same thing for this chemical stain. I'm not sure about that. I'm looking around right now.
Okay.
But we'll just say it's something like that.
Okay.
That's kind of vaguely how it works, right?
Yeah.
So I tried to poke holes for a while in their anoxic thing. I think it stands. Yeah. I'm convinced that it's an anoxic region.
Okay.
So then you say it is an anoxic region. What's left?
Yeah.
Right. And the answer is glycolysis. And so then with that answer in hand already, they go to spatial transcriptomics, which is where I get suspicious.
Oh, really? Okay.
I love spatial transcriptomics. It's the coolest thing in the world. Spatial transcriptomics has a lot of problems with it that are going to be a lot of problems for a long time. One of the problems with it is you have, 20,000 genes in your bird, and it's going to report spatially the intensity of how much RNA you have for each of those 20,000 genes to some extent. Some genes it will work very poorly on. Some genes it will work terrifically on. So if you have one gene way higher than another gene, it doesn't really mean anything, right? Because you don't know what genes it's going to work terrifically on and what genes it's going to work terribly on. So for instance, when I do spatial transcriptomics, I'll be like, oh, I'm really interested in this particular kinesin. I'll look up my data and I'll find for all, 1,000 pixels that I didn't get a single hit on that kinesin. It doesn't mean the kinesin's not there.
Yeah.
It means that the system isn't very good at picking up that kinesin.
Yeah.
I might get, however, 1 million counts on some mitochondrial protein, right? It's just good at picking up that mitochondrial protein.
Yeah.
So you have to look at spatial differences in
a single gene.
In a single gene, which they do. But then you have a problem of, are you looking at spatial differences where you're picking up 1, 2, 3 counts? Are you picking up 100, 200, 300 counts? And how do you average together your experimental replicates? So here they have 4 experimental replicates of 4 birds that they've done this to. And they need to average together those spatial transcriptomics. They never have the same slice, the same cryo slice of the eye. So they're never looking at an image that looks particularly similar to the other images. And it's really a struggle of how do you put these data together to get systematic information. And also, you want to tell a story about glycolysis, but maybe the real story is actually about the mitochondria, and you're going to miss it because you didn't get the mitochondrial sequence. Because the problem with spatial transcriptomics is you don't know what you don't know. That's
that problem resonates with me because it's the same problem with mass spec, the mass spec that I've done, which is like you get the peptides that fly well.
Yeah.
And
exactly. So what they do here is quite interesting. They model, and I think they model this via the hidden mitochondria data that they never show you. So get a little bit suspicious here. They model what the oxygen levels must have been, I think from the mitochondrial staining, and they normalize the spatial transcriptomics to the modeled oxygen levels. So they model a region that they say is going to be roughly anoxic, and they say, what was the glycolysis expression here? What was GLUT1, which is a glycolysis transporter, glucose transporter? What was GLUT1's expression at 0 partial pressure oxygen as modeled by the mitochondria data that they don't show anywhere? This is a clever solution.
So, sorry, for figure 2, like B through D, they're saying these are like Go terms.
I'm not a fan of
clusters
Go terms.
I think many people are skeptical of Go terms or feel
they also show in H, they use 2DG.
What is 2DG?
So 2DG is a version of glucose that can't be used by the cells, so it just kind of sticks around. what it really shows you at the end of the day is where glucose is being transported to. I essentially just ignored this data. I don't think it's bad data, but I've taken a lot of 2GD data myself, and I essentially just ignore it for myself as well. Because at the end of the day, I don't have like a strong intuition of what it's really telling you about the physiology of the system. Okay. It's kind of telling you where glucose goes when the glucose can't be used, which I don't think is super. The big heavy hitters is figure 3, where they show you, hey, we have GLUT1 transporters in pecten. I would like to see GLUT2 and GLUT3 for the record. I know you don't know what you don't know, but I would like to see what it looks like.
Yeah.
The thing here is right there, like, we know that there's no oxygen at the retina. So we know that there must be glucose getting in here somewhere, right? And then here we've identified a pipe that glucose can flow through. Therefore, glucose is flowing through this pipe. It might be wrong, right? It's like saying, like, I have some water uphill. Here's a pipe that water could go through. Therefore, water must be flowing through this pipe, right? That's not necessarily true.
I thought they did some labeled glucose thing.
Yeah, that's the 2DG stuff that I don't think has any super big meaning.
Okay.
And even if I did believe in it, I wouldn't be convinced by this image.
You can't like watch, with this I forget what the acronym is that you keep saying.
2DG.
2DG?
Yeah.
2DG.
Yeah, unfortunately we are for once in my actual element where I have actual opinions about the experiments.
For the 2DG, you can't add it and then watch it?
They certainly can't, right? They've got a whole freaking bird. They can't do live microscopy. But again, it's not usable glucose, right? So what is it really? And in fact, like, you can kind of use it as a drug to inhibit glycolysis is what it's often.
But you can't put in, I guess, what I had thought. And
do, if you could do microscopy for your system, which they can't
it's a good point.
If you could do microscopy for your system, there are these like glyphin green reporters, which will report glucose density or glycolysis activity. So there are fluorescent reporters. They're very tricky to use. I think I would believe it if I saw it. I think many people have tried it and been like, this isn't working, including myself.
Okay.
But again, this is a whole bird, right? Live imaging is off the table, at least that way.
Okay, so your complaint about the spatial transcriptomics is that they rely on their knowledge already that the tissue is anoxic. I
think it's terrific that they did spatial transcriptomics. My complaint with spatial transcriptomics, not for this paper, for all papers, is that I think you can almost always tell any story given spatial transcriptomics, right? My paper that I'm putting out, I could have just showed one microscopy image and the spatial transcriptomics data. It backs up everything I'm saying, right? But because it's so sparse, I'm finding when talking to other people that everyone can back up all of their stories with spatial transcriptomics always. You have 20,000 fields to pick from. One of those fields is going to say what you want it to, right? I know it's not always true, but it really is such broad data and such low numbers on the data that I think it's very easy to say what you want. So for instance, I'm not sure how they've normalized, but like look at figure 3b. They have these numbers 0, 1, 2, 3, 4. Yeah. I'm not sure, depends on their technique, but it is possible that they literally mean 1 count of RNA detected in this pixel, 2 counts of RNA detected in this pixel, 3 counts of RNA detected in this pixel, right? Those are incredibly low numbers to say that you're sure about something, right? If you took a image of something and you had 4 photons at the highest density of photons, you wouldn't bother thinking about that image.
Can you not do IF on this?
Well, not only can you do it, they did it.
Oh, they did it.
That's figure 4. Yeah. And most frustratingly, they did IF for mitochondria as well. They're actually basing most of their data off of some model, and they're not showing it, which makes me... Again, I'm not suspicious. I think the thing that makes me so mad is I really think that this is a seminal paper, that this is a paper that I will talk about for 30 years, that I'm really happy that I read this paper. That's why I'm so mad that there's a thing wrong with it, is that I think that this is a huge, terrific paper, and why I blame the editor. Do your job. You have a job. If someone says there's no code to report in their paper and they're showing anything other than, like, what did the editor think? That this paper was made with like sticks and rocks? If you say that there's no code in your paper.
Yeah, I do think this is the editor's job, but the code, like the language in the Nature checklist. So I'm going through the Nature Cell Biology checklist, author checklist right now. And the code is like, only if the code is important for the finding of your work, or like And so I was like...
Code availability in the methods. No custom code was used in this study. All analyses were performed using standard software packages as described in the methods. In the methods, if you follow it, you can find a secondary piece of supplementary information that is not on the main thing, in which they describe a really long Mathematica notebook that exists somewhere.
Again, the code, what you're supposed to talk about in the code availability statement is murky. And so I could imagine somebody saying like, oh, it wasn't central to their paper, so we don't actually need to report it.
Makes me even angrier in nature, right? If it's a systematic problem and it wasn't like one editor's mess up, but like it's all editors' mess up.
So I think the point of the checklist is to try and prevent this.
But the editor should have editor checklist. Is it a paper? Was it entirely crafted with sticks and rocks? No? Get the GitHub for the code, right? Like the default should be, give me your GitHub for the code. There doesn't need to be a checklist for that. The editor should do a Control F for GitHub on the paper, right? If it's not there, alarm bells should start ringing. And there do exist. There exists for sure papers that are literally written without code. But this ain't one of them, and there's not going to be many that make it to Nature that are literally written without custom code.
You can put a GitHub in and not put anything in the GitHub.
Sure.
Right?
Then I don't blame the editor. Then I blame the scientist.
Yeah.
But the editor better do a Control F for GitHub, especially if you're Nature. Like, this is the major leagues. This is a seminal paper. Come on. And anyways, they show immunostaining in figure 4 for vimentin and GLUT1 and MCT1. You can decide if you think that GLUT1 staining is very convincing.
Again, I'm just not super oriented.
So the top is not anoxic, and the bottom is anoxic.
Okay.
So this GCL layer is the anoxic layer.
Okay.
And that layer is the layer that has bright GLUT1. But, the way immunostaining works, it's very much just like food coloring, right? If you put some food coloring on your finger, right, it'll make the top of your finger red. It doesn't make the inside of your finger red very easily, right?
They're not slicing the other way?
Well, that's a good question. Are these cryo slices?
I would have assumed that they were slicing like perpendicular to these pictures.
Yeah. I think you're probably right, in which case my opinions are not that valid here.
I mean, I agree that if what they're doing is what you described with the mito tracker, that this could be an artifact. But if it's a slice and an immunostain, which is what I had wanted from the
immunolabeling of GLUT1. Zebrafinches were euthanized by decapitation. Always a terrific start to a methods section. God, Jesus Christ. I'm just going to keep reading because it gets worse. Eyes were enucleated and the anterior part of the eye was cut off with a razor blade. The vitreous humor was gently removed from the resulting eye cup, which was then fixed. Okay. Going on, they embed an OCT. They make vertical sections 25 microns on a cryostat. So yeah, they're 25 micron sections. So I take back the purple line at the bottom. It seems like good immunostaining to me. Yeah. That there's more GLUT1 on that layer. I'll believe it. I would like GLUT2, GLUT3. It doesn't mean anything that they're not there, but just like, why do I only have one glucose transporter? And I think, unfortunately, probably the reason why they're only showing one is they did the other ones and they didn't see anything.
Yeah, is the spatial transcriptomics data like deposited somewhere?
I believe so.
Yeah, but I mean, I wonder one thing is that they could have they looked in the spatial transcriptomics for GLUT2, GLUT3, and there weren't enough counts to say anything. And so they were like, oh, it's not worth doing the immunostaining. But...
Well, that would have just made me more interested in the GLUT2 and GLUT3, that you didn't get it by spatial transcriptomics. So you don't know if you got it or not. Okay. So you really, really should do the data. They say that it's available on Geo. But yeah, in theory, you could go check this and learn about the GLUT. Maybe I should do that because I could use it to look at the mitochondria.
Patterning. Yeah.
Yeah, and then they show, and I think that this is kind of fun, like this is where they really go as hog wild. If they do figure 5 and they're like, now we're going to do everything we did, but for all the creatures.
Yeah.
And they do an anole, they do a pond turtle. I was complaining, we did a paper before where they did just the chicken and then they made it look like they did all these other creatures. And I was like, come on, do all the other creatures. I did all the other creatures. You got to do all the other creatures. Here, they did all the other creatures. And the husbandry, there's so many in the methods. There's so many little gems of how they did things. All animals were purchased from local dealers and housed at our house for at least our house being the city in Denmark, not their house.
Yeah.
Our house for at least 7 days before experimentation.
Wait, but this plot that you complained about in figure 1 whatever, 1F.
Yeah, now it shows up in force in 1B.
Yeah, so were they doing mitochondrial imaging in all of these eyes?
Yeah, I think so, if I understand correctly what they're doing. And it's also how they align their spatial transcriptomics. It really does seem like a very linchpin bit of data that they're not showing at all.
And it's not in the extended data. I mean, I assume you looked
for it for a long time. I texted other people about it. I emailed the author. The author got back to me, but they misinterpreted what I was asking for. So they still might get me that data. That being said, they did it in all the animals to see, hey, I had a question when reading this. This is true for the zebrafinch. The zebrafinch flies.
Yeah.
They make this argument, hey, when you're flying, you don't have that much oxygen up.
But you still want to be able to see.
But you still want to be able to see. So they've got to change how their eyeballs work. My question was like, hey, chickens don't go that far up. So they do chicken. The chicken eye works the same way as the zebrafinch eye. They do either an alligator or a crocodile, which is the next closest thing to a bird. And that already doesn't do this. It has the normal eye setup. The turtle and the anole already, none of these have anoxic regions where there's no oxygen in their eye. So really, it's just the bird clade that does this, which is, I think, super fascinating.
Yeah, it's very cool. Did you catch the fun fact at the beginning that the naked mole rat's brains?
Yeah, I highlighted that for you. Yeah, I highlighted that for you.
It made me think about, like what makes a tissue anoxia tolerant and like what's different about this tissue, like because it never sees oxygen. I mean, obviously it's different in its metabolism, but yeah, like the question you asked about, like, are there mitochondria and what are the mitochondria doing there? And what's different about this tissue so that it, as a totally anoxic tissue.
And, this is what I'm all about, is that there can be patterns of energy metabolism in tissue that can tell you totally new things about how that tissue is working.
Yeah.
I feel so much richer for having read this paper, for knowing more about bird's eyes. Like, I've been talking, I think I started asking people this in 2018 of, do the two different bird eyes have two different refresh rates? They have like a prey eye and a predator eye, right? And I've always been confused about the refresh rates of bird eyes. So just having a little bit more information about the nitty gritty of the bird eyes. I'll forget all the specifics. I always forget all the vocabulary for eye structure.
Yeah.
I every time I go to the glasses, I went through this all with my eye doctor just a month ago, where he took a picture of this thin part of my eye and where the vascularization is. And we sat there and I would point and be like, what is that part called? How did this part of the lens work? And we sat there like for 30 minutes as he walked me through it all. But every time I do that with him, and every next week I forget the whole thing. So I'm not so good with the eye terminology, but
I got to do some experiments, like more experiments with eyes, and maybe it'll stick.
I don't have the permission to do this, but I would love to do some adult chicken eye staining of mitochondria.
Oh, because you don't have the eye cook for a...
Right. This would be a 10-year-long nightmare to do at most universities in America.
Interesting.
Yeah.
That's unfortunate.
But maybe you could get because it's the eyeball, you can maybe you could get a mitoreporter chicken and you could image the adult chicken without killing it just by looking into its eyeball and looking at the mitochondria at the back in the retina. Now that is an experiment I actually want to do. Is make my mito chicken, get the adult chicken and look in the back of its retina.
But then you have to get like a whole optometrist's eye thing and get the chicken to like put its eye up and...
Yeah, that sounds brilliant. Doesn't that sound like a blast to do? You train the chicken, like put your eye up to here for 30 seconds and then you'll get a treat, right? And anyway, that sounds like a lot of fun to me.
Sounds like a pain to me.
Actually, there's an easy way to do this, right? There's the way there's a bug with the chicken, right? There's like a defect.
Yeah, that you can like get their heads to flop a little.
Yeah. So you can do this thing where you draw it in the sand. The chicken has like an error message. It just won't move for like a minute. And then you can take your measurements while it's doing that. So you don't even have to train the chicken.
But you do have to like have the lens, like the camera lenses set up so that you can image the eye, right? Like they have a really fancy way to image your eye at the eye doctor.
Yeah,
right?
yeah. So you have to create that. Yeah.
Yeah, let's see if I highlighted anything else that I wanted to talk about. I mean, I don't know what a Mueller cell is.
Yeah, so they go into this at the end. They're essentially like, how does glucose get here? And how is the lactic acid being collected? And so they say there are these Mueller cell-mediated nutrient exchanges, right? I don't think that this is super interesting, but if you're publishing something at this level, you have to come up with a mechanism by which things work, right? So the question is...
And so this is like a special kind of cell, or it's just
this is a special kind of cell. Okay. that they identify by MCT1. That's why they're doing this MCT1 immunostain.
What is MCT1?
It's just a gene that Mueller cells tend to express a lot that other cells are not expressing a lot.
Okay, so Mueller cells were already known to be a kind of cell in the eye.
They're definitely not calling. They haven't like, and Mueller cells usually don't have like really high expression of GLUT1 or something, right?
Okay.
And these ones do. And they say these Mueller cells are specifically adapted to do this glucose transport and lactic acid uptake. Cool.
Yeah.
I'm not going to not believe that. Something's got to be doing it. I just don't really care what is. It's not my thing. I'm sure other people are wild and think that's like a linchpin of the discovery.
Yeah, for me, like, I thought this was a cool paper. I thought it was cool from the title. Like, it was really well titled. You got the message very early on. And for me, that's kind of all I needed because I'm not in this field. I'm not going to be following up on these experiments. And so it was like, yeah, I got a lot from the first half of the paper and very little from the second half of the paper.
I'll say good year for bird physiology, right? There's a lot of bird physiology papers that have come out this year that have like moved forward how much we know about birds.
Yeah,
it's an interesting endeavor that kind of naturalists are going off on.
I feel like birds are very much in the popular zeitgeist these days. A lot of bird watchers, a lot of people playing Wingspan.
If you could add one thing to this paper, what would it be?
Yeah, because it's conserved across so many birds, is there not a bird that we have good transgenics in?
No. I mean, there's nothing that we have mitochondria in. The best bird that we have transgenics in is the quail, I would say, or the chicken. But either way, you don't have any of this stuff. What you could do, again, because it's an eyeball, is maybe you can do 2 photon on a living bird. I don't know how reasonable it is to ask for any of this stuff.
Yeah. No, it's definitely not reasonable to ask for a
And I don't know if there exist bird eye organoids. I know there are eye organoids, but I don't know if there are bird eye organoids. And I would assume no.
Yeah.
Okay.
Yeah, it's an interesting problem how to study something in like something so big in a living organism. I know this is something you think
this is my favorite problem. This paper is just my jam. Everything about this paper, man, I hope the authors don't hate me for this one thing. Because man, authors love your work. This is terrific.
I do feel like it's a very Bez paper. Like there's a lot of your creative writing is like resonates with this.
There were parts of this paper like, the pecten oculi. I think I've written a short story about that. For Asya, why was your dad so into this paper? I was trying to, at this point, make this like spatial energy metabolism readings like my career linchpin, that using techniques like this that they've shown in the bird, like using a needle that you poke through and see the oxygen levels at different parts of tissue, you could use techniques like this, really 2 photon techniques like this, as diagnostic devices. That's the spatial energy metabolism part. The eyeball, I mean, a question that you ask yourself when you're teaching these like physiology courses, we ask the students, what is your favorite organ? Right? So Linnea, what is your favorite organ?
I've never thought about it before.
Okay, top of your head.
I guess the heart.
The heart, yep. Heart's a super cool one. Heart strings are super interesting. My answer to that was the eyeball. And I said in the same breath, but I would never study the eyeball because it's so hard to study. And then I've gone on to study the eyeball a little bit. And man, it's a blast. It's just a beautiful organ. If you like physics, man, the eyeball is a lot of fun.
Do you think
that
this
neuroscience?
is... Oh, sorry. I'm interrupting the Asya time.
Oh, okay. Yeah. So I got really interested in the eyeball for diagnostics. And then the last thing about this paper, the trifecta, is that it's birds. And yeah, why was I so into birds is a hard thing to explain. We had these chickens as pets, and so I became really, really deeply interested in their physiology. As every pet owner does, they learn about how their pet works and how it becomes sick and how it becomes healthy. And so I was really interested in the physiology of birds, but also I started doing experiments on birds. The tempo for the experiments on birds is very nice because you're just cracking open eggs. You get 100 eggs a week. I was thinking about it. I calculated out. I think I've done 10,000 embryos during my postdoc. And you get to do experiments really fast. But the biggest thing about the birds is I started to meet other bird researchers, zebrafinch neuroscientists, chicken embryologists, quail physicists who are studying gastrulation on the quail. And I loved each and every one of them. Like the bird community has significantly extended the period in which I'm interested in birds because the bird people are so terrific in a way that I haven't really encountered since I was an undergraduate in physics and meeting physicists for the first time. So I think it's not a surprise that we're doing this terrific spatial energy metabolism in this terrific organ in birds first. because the bird people tend to flock to the same questions that I love. That's why I'm so about this paper. Yeah.
Okay. Can I ask a science question?
Sure.
Before we sign off?
Sure.
Do you think that this anoxic tissue, how does oxygen work in the embryo?
Yep.
So do you think that this part of the tissue, or is the whole embryo anoxic? And if not, then do you think this pattern also exists in the embryonic?
This is a terrific question. A lot of people's objection to my research program when I started it was development is largely a hypoxic environment where there's low oxygen. To what extent that is true, I think is like unclear. Like to what extent if they're not oxygen present, I think it's unclear. And it's unclear at many scales. Like because the vascularization has not come online, even if there are regions that are not hypoxic, there might be other regions that are hypoxic. Are there regions that are anoxic? I don't know. To be anoxic, you need mitochondria to use up the oxygen that are there already. We have seen through my experiments that there are a lot of mitochondria and they're doing a lot of stuff in development at many different time points in development. But to what extent the embryos are ever anoxic, I don't think I know or we know. It's a super interesting question. For bird development, especially early bird development, you're right at this air interface on the top of the egg, right? The bird grows on the top of the egg.
Yeah.
So you're exposed to a lot of air and the egg is oxygen permeable. For mammalian development, you're inside another creature. The other creature is not all that oxygen depleted, right? And so they can provide you oxygen in many ways. But again, you're not vascularized. I think before I did my experiments, if you ask AI, the AI would say it's all hypoxic. Some of it might even be anoxic, and mitochondria aren't doing anything in any of this tissue. My experiments have shown me otherwise. And if people believe my experiments, they will also believe otherwise. I know that my experiments are true, so people will find over the next 10 years that there are tons of mitochondria doing tons of oxygen consumption in these early developing systems. So they're not anoxic, but maybe they're still slightly hypoxic. Yeah.
Since there seems to be oxygen in a lot of metabolism, do you think that this pattern of I guess it's totally different because the eye. So is there a point in the embryo where the eye has already developed? Like where it's a past tense and now it's developing other things? I just wonder if you could see this pattern in an embryo.
But see what pattern? Because here we don't know what the mitochondria look like here.
Oh, but you could look at GLUT.
Oh, see if the GLUT1 pattern.
Yeah.
But it must be. It had to have developed into this pattern, right? So
what are you talking about? The cells express GLUT1, right?
The cells express GLUT1, right? But that has to do with their fate, right? At some point, they've got this GLUT1 expressing fate. And that has to happen during development, not post-development.
That's a fate thing? No, you can...
Oh, you're saying that the cells are upregulating GLUT1 as a response to their environment?
Yeah.
That's a wild theory. That's crazy.
No?
And you're saying that actually if oxygen did flood the eyeball somehow, the GLUT1 levels will go down.
Yeah, they show that?
Do they show that?
Isn't this one of the things they do is they flood the...
They do flood the eyeball or they flood the creature with oxygen.
But maybe they only do the oxygen probe measurements after that.
Yeah, I sure as hell don't think that they do. Like, how would you even do it? Because it's spatial transcriptomics. They have to fix the creature. So you're going to fix the creature while it's at high oxygen? Maybe. You keep it at high oxygen for 3 or 4 days.
Okay.
You fix it. That's the experiment I want. No, really, because you've created such a terrific question. Keep the bird at crazy high oxygen levels for a couple of days.
And see if this pattern goes away.
You fix it, do spatial transcriptomics, and see if the GLUT1 is still highly expressed at the retina. I love it.
Yeah, I think you should do some modeling. Someone should do some modeling before doing that experiment for thinking about how high oxygen you would need at that retina, at the edge of the retina to get oxygen into the middle of the retina.
You know who's already done that modeling?
You.
They have. But you don't have their code. So you can't see what they did. Probably said that too loud. Great last question. Man, I don't know if anyone's going to listen that far in, but that's such a good way to end. But we were dreading actually recording this. I think this is the first one we were ever not jazzed to record.
Yeah.
And my mood is so much better
now.
Like, this is still the highlight of my day is recording this.
Yeah. And we did nothing today. So this is so nice to have recorded it and have read a good paper and talked about it with you. So.
Okay. So on that note, you want to take us out?
Thanks everyone for listening. Bye.
Bye.